Alright, ladies and gents, we're going to start our topic of macromolecules today. So, let's break this word apart for a second. While molecule, hopefully you know, is some sort of building block for a compound, two or more atoms joined together. Macro, we're talking big. So we're talking these really large molecules. And they're organic macromolecules, so giant, huge molecules that are joined together by these smaller molecules, and they're really big necessities for living things. And when we classify these particularly large molecules that living things need, they end up in four main classes. So your carbohydrates, your lipids, your proteins, which hopefully you recognize from the foods that you eat, as well as nucleic acids, which are things like this. Hopefully this looks familiar. This is DNA. So we're going to talk through the major classes in this particular section, some of the things you're going to be expected to know, what are their building pieces called, what are the big molecules called, what are the types of specific bonds that hold those pieces together, elements that they're made up of, what's their function. And so that's really going to be the big focus for this particular section. Just like I mentioned before, flashcards are really kind of be your best friend. So make sure that if you're not using the Quizlet set that's all posted on Schoology for you, that you're making your own somewhere. Because this unit is really a lot of memorization and identification. So let's jump into some basics first. So before we even get into those four main classes, we need some vocabulary to talk about what macromolecules are, how they function, how they get formed, how they get broken down. So two big terms that you need to know are the word polymer versus monomer. So poly means many, right? So a polymer is this really long molecule. This is essentially where that macromolecule term comes from. Those polymers are made up of lots and lots of similar or potentially identical repeating units. And those units that build up those polymers are the monomers themselves. So these are your building blocks. So sort of think of a polymer being this big Lego tower that you've created, and the monomers are all those individual Lego blocks that you had to put together in order to make that big tower. It's essentially how this works. So monomers are all these building blocks or building pieces that we stick together, bond together chemically to ultimately make a big long polymer out of it. There's also some chemical reactions that you need to be aware of that are involved with the creation and the destruction of those polymers. And this is where water comes in. So first off, if I'm forming a polymer, I'm making the polymer longer, I'm making it bigger, I'm adding Legos to that tower, if we use that analogy. We're talking about a chemical reaction called dehydration synthesis. And I'm dehydrating my monomers in order to synthesize or create a new bond. Alright, so that's where that word comes from. So there's the dehydration part. Water's coming off. Very clever, biologically speaking, that all of our monomers, regardless of what type they are, have an OH and a hydrogen somewhere. So those guys are going to come off. They're going to create this space, this blank bond now, when water leaves, that I can now plug those guys together. Alright, so dehydration, just like if you're dehydrated, you're lacking water. Well, that's what I'm doing. I'm pulling water out. And synthesis means to create. So I'm creating a bigger chain. That's where that term comes from. Heads up, you may also come across it as a condensation reaction. Same thing, just different word. Condensation also involves water, and so that's where that comes from. But I find that for most kids, this term, dehydration synthesis, makes more sense. And it's also the one that gets used a little more commonly. So that's the one we tend to stick with, but want to throw in that condensation just in case you happen to come across it somewhere. So dehydration synthesis, I'm making a polymer. I'm making it bigger. I'm adding monomers to the chain. In order to do that, I've got to take water out. Well, guess what? If I'm going to break down the macromolecule or digest it, if we want to think of it that way, and make more monomers out of it, I'm going to add water. So it's essentially the exact same process, but backwards. So here's my water molecule. That water is going to come in. It's going to break this bond. One hydrogen from the water is going to go this way. The OH part of the water is going to go that way. And I've now broken the chain into smaller pieces. And we talk about this as a hydrolysis reaction. Well, if we know our prefixes and suffixes, hydro means water, and lysis means to break. So I'm adding water to break the bonds. This is part of why water is so important in ensuring that your body can digest and use all of that food that you take in on a daily basis, because without the water, these chemical reactions don't happen, which means your body's not able to break down the food that you're eating, nor can it then use those building pieces to create other molecules that your body needs to function every day. So your metabolism essentially shuts down because there's no water. Right? You could be stuffing your face with food, but if there's no water content to that food, these processes can't happen, and it really doesn't matter. So let's get into some of the classes of macromolecules, and we'll start off with one of my personal favorites, carbohydrates. Alright, and you're going to find that all of these macromolecules, they're coming from your diet. That's where you get them from. And then your body's going to break them down, digest them, get them into your cells, and use them for other processes. Maybe it's for energy, maybe it's to build new molecules. But it's all got to come from somewhere, and because we're heterotrophs, we can't make our own food. That means we've got to consume it from somewhere else. So, for us humans, where do most of our carbohydrates come from? Well, they can come from simple sugar molecules that we find in things like your soft drinks, your candies, or they can be in these more complex polymer structures, things like starches, so lots of pasta, potatoes, pretty much everything you eat has got carbs of some level, unless you're talking meat. So check out those nutrition labels and see just how many carbs you're taking in. You might actually be surprised some of the things that you find carbohydrates in you wouldn't have expected. But they exist, so they're there. What do we need them for? Well, you definitely do need them, alright? They are your main source of quick energy, so this is what your body's going to break down first to make sure that you're getting enough energy to do all your cellular processes, all those life processes that you need to do to function every day. If we don't use it, well, then we can kind of use some of it as storage, or eventually it gets put into long-term storage and we end up making fat out of it. And in plants in particular, they also use it as a building material, so they're actually able to create their own carbohydrates via photosynthesis. And so they'll also use it for some of that energy, some of that storage, but they'll also then convert some of it into those building materials as well. So definitely want to know the function of carbohydrates. What about some of the chemical stuff you should know? Well, structurally, we got three major elements that carbohydrates are made out of: carbon, hydrogen, and oxygen. And when we take a look at carbohydrates, the quickest way to pick out from a chemical formula that you're dealing with a carb is that you've got this 2-to-1 hydrogen to carbon ratio, or sort of this general molecular structure that you see down here. So think of them like math class, right? So if I've got n number of carbons, then I have to have double that for my number of hydrogens, and at least assuming we're talking simple carbohydrates, monosaccharides is the term we'll use in a minute, then your oxygen will also have the same number as your carbs, or excuse me, as your carbons. You start getting into your polysaccharides, and that's going to get knocked around because of the waters getting removed, but at least that general molecular formula follows for your simple carbs. What if I'm looking at a chemical name instead? Well, then these guys are going to end in OSEs. most of the time. So things like glucose, galactose, fructose, right? Those are all carbohydrates. So OSE, if you see that, then it tells you that it's a carbohydrate. So speaking of that carbohydrate formula, let's do a couple of practice problems here with that sort of general molecular formula. So if I've got 6 carbons, 6 oxygens, all right, remember it's CN, H2N, ON. So if I've got 6 carbons, that tells me that I end up with 12 hydrogens. Now I'm going to have you pause the video here, try and answer those last 3 yourself, and see how you do, and come back and check your answers. Alright, so if we double check, next one's also missing the hydrogens, so it should be a 2N kind of deal, so that means I've got 10. Last one, I've got my carbon, my hydrogen, and if we're assuming this is a monosaccharide, then I should have 4 oxygens. And number 4, same deal, let's assume it's a monosaccharide, so my carbons and oxygens should be the same number. So if H is 2N, then that means both the carbon and oxygen number have to be half of that, and that's where the 7 comes from. So speaking of monosaccharides, these are the monomers for your carbohydrates, and you're going to want to make sure that you know that term. They are the simplest of our carbs, and they are actually, interestingly enough, the only monomer we're going to talk about in this entire section that can actually function by itself. Alright, all the other monomers that we're going to talk about for other categories, they need to be combined with each other to make some bigger structure in order for them to actually be biologically active, so that they actually do something in a living system. These guys can actually work all by themselves as simple sugars or monosaccharides. You should also recognize some of the functional groups that we see in our carbohydrates, and there should be a blank spot in the picture in your notes to add these. So we've got hydroxyl groups going on here. We've also got carbonyl groups going on, and just some examples you should be aware of. Things like glucose, okay, that's a monosaccharide. Fructose is the simple sugar found in fruits. Galactose, simple sugar found in milk. So all sort of have the same basic ring structure, just some of the carbon groups and things that are coming off are going to be a little different, and that's what's going to set them apart as far as this goes. Or the number of carbons that they have in the actual ring itself can change. But generally speaking, they've all got a ring structure that looks something similar to this. Now if I start putting monosaccharides together, the first thing I'm going to form is a disaccharide, and di means 2. So a disaccharide is 2 monosaccharides joined together. It means a single dehydration synthesis reaction has happened, so I've removed the water, and I've been able to link these guys together. And so I get this particular covalent bond that forms between these guys, and that's something called glycosidic linkage, and you do want to make sure that you know that term as well. And I believe there's a blank in your picture in your notes to add that in as well. So if I go ahead and make sucrose, which is a disaccharide, it's a glucose, via 1 glycosidic linkage because of a dehydration synthesis reaction, plus a fructose, I get glucose. This is your table sugar that you're used to using to cook or bake with at home. Okay. Maltose, or what we know as grain sugar, is a glucose plus a glucose. Lactose, this is what people are typically unable to digest if they're lactose intolerant. It's your milk sugar. It's a glucose plus a galactose. So that's how we sort of start putting these together. Technically speaking, disaccharides are not polymers for carbohydrates. They're just not quite big enough. But they're not really monosaccharides, so they're not really monomers either. They're sort of in this weird flux in between. Just something to be aware of. Okay. But what do you really need to know? Well, disaccharides are 2 sugars. Your true polymers are your polysaccharides. So these are really long chains of monosaccharides linked together. You'll often hear them from a nutritional standpoint as complex carbs. So things like starches. These are the big storage molecules that plants use for the excess glucose they produce via photosynthesis. And it's what we typically end up eating. So it's those grains that we find in our breads, in our pastas, in our potatoes. Okay. Cellulose, this is the major structural component of your cell wall. So plants will take some of the glucose that they create via photosynthesis, and instead of storing it in starch, they'll use it in cellulose to build new cell walls as they're making new plant cells. In terms of us actually eating it, we can. We don't really break it down very well, though, but it's what we talk about as dietary fiber. Okay, so make sure that you're able to properly use the restroom on a regular basis. Alright, you need that fiber in your diet, and it's going to come from cellulose. The biggest one that most kids recognize or can see in terms of cellulose in the foods they eat are celery. All that stringy stuff. That's a bunch of cellulose. In us, we also can store, at least short-term, excess sugar in our body, and we store it in the form of a molecule called glycogen. So glucose is in your body, that's what you're building up, that's what you're utilizing on a regular basis, but if you don't quite need it all, your body will throw it into short-term storage in the form of glycogen, and paying on that to that into your liver. If it stays there long enough, eventually your body will turn it into fat. So structurally, if we're talking polysaccharides here, they're repeating units, right? So in this case, I've got 3 different carbohydrates or 3 different polysaccharides. They're all glucose molecules. So what makes them different? Well, starch is the first one, and it's just this long chain of glucose. Glycogen, this is what we use in our body. Alright, similar idea, except you get these long chains that kind of pop off of them. Alright, and so structurally it ends up being a little bit different. It twists into this helix, but then we've also got these side branching chains as well. And there's your cellulose. So these really long, stringy chains that kind of end up almost like thread in yarn, right? Okay. So although they're all made up of strictly glucose, the way that they come together is different, which is what makes it a different molecule. Lipids are another category, and lipids are found in all sorts of things that we consume on a regular basis. Animal fats, so the fat in bacon, for example. Fish oils, plant oils, so things like olive oil. Your butter and your shortening. Okay, margarine, that's all lipids. Nut oils, like the peanut oil here. Alright, those are just some examples of where we can get lipids in our diet. What are they there for? Well, they are now long-term energy storage. Okay, so glucose is quick. You're using it relatively quickly after you consume it. Your lipids, however, are the ones that are going to get stored a little longer. And although when we break them down we get more energy, this is not typically the first thing that your cells go after. Also there for insulation and cushioning of your organs. You do need some fat. It is necessary. Without it, you're losing some of that cushioning of your internal organs, which is not necessarily a good thing either. Of course, you can have too much of it, alright, which also causes problems. But some fat is appropriate. Also going to be a major structural component to your cell membranes. And we also have them there to help aid in the absorption of certain vitamins. So some of that other material that our body needs, your cells can't take it in without the lipids being present. So once again, why do you need some of it? Well, there's why. So chemically, what do you need to know? Elements, still the same. So carbon, hydrogen, oxygen. So how are you going to tell the difference chemically if I gave you a chemical formula between carbs and lipids? Well, you're going to look for that carbohydrate ratio of number of carbons to number of hydrogens, right? So for every one carbon, there's 2 hydrogens in a carbohydrate. You won't necessarily see that ratio in a lipid, so that's going to be a key right there. Chemically speaking, lipids are interesting in the fact that they are hydrophobic. So these are things that are not going to be soluble in water, and think about it. You've all put oil in water before, you know that they don't mix. Okay, so unlike things like the sugars or the proteins, these guys are not going to dissolve readily in water, nor do they have a true polymer. So unlike every other category we'll talk about that has a true monomer polymer structure, your lipids don't. Okay, they've got some building pieces, and you should be aware of what they are and how many exist for each type of lipid, but there really is no true monomer polymer structure in the sense that I've got these repeating units that I'm sticking together to make a bigger chain. But they do typically have a glycerol molecule plus a certain number of fatty acids. So which kinds of lipids should you be aware of? Well, here's 3 categories that you should know, and we'll talk about a 4th a little bit later. Waxes, so things like the beeswax in your candles, right? So I'm talking a single glycerol plus a single fatty acid. Not going to spend a whole lot of time talking about them, just to be aware that they exist. Fats and oils we'll spend plenty of time on, so what we're dealing with over here, your butters, your margarines, your shortenings, your oils. Single glycerol plus 3 fatty acids, and then your phospholipids. And we'll spend lots of time over the course of this year. These guys are going to keep coming back. And they are 1 glycerol plus 2 fatty acids. So let's talk fats and oils first and talk a little bit about structure. So these are a glycerol plus 3 fatty acid molecules, also known as a triglyceride. So if you've ever heard anybody talk about triglyceride levels because of health issues, this is what their doctors are concerned about. How many triglycerides or how many fats and oils are they taking in a given day. And all of these guys go through a dehydration synthesis reaction at each connection point here. So 1, 2, and 3. Which means when I'm going to build these molecules, every time I make a complete triglyceride, I've got to actually remove 3 waters, 1 for each connection to that fatty acid to the glycerol. And so, just like there was a bond type you needed to know in carbohydrates, well, there's one here as well. So we make these covalent bonds here, but in your lipids, fats and oils, and specifically we talk about them as ester linkages. Alright, so glycosidic in your carbs, ester linkages are going on here in your lipids. We can also distinguish your fatty acid tails from each other, and this is really where a lot of the properties of your lipids come from. So first off, every fatty acid tail ends in a carboxyl group, so there's another functional group you should recognize. Okay, but then I can also take a look at what's going on in the chain of carbons that's coming off there. So beyond that carboxyl group, I've got a hydrocarbon. And we were talking about hydrocarbons in the last section. So if I'm dealing with a saturated hydrocarbon, or in this case specifically a saturated fat, then that means that that carbon-to-carbon chain is maxed out with the maximum number of hydrogens possible. So in order for that to work, all of those carbon-to-carbon bonds have to be singles, right? Just like the definition of a saturated hydrocarbon we were talking about. And so as a result, you get these straight-chained fatty acid tails that are actually able to stack really nice right on top of each other, real nice and close. And so saturated fats are things like your animal fats, butter, and these guys are typically solids at room temperature. Flip side to that, though, are your unsaturated fats. So now I've got less than the maximum number of hydrogens because at least somewhere I've got one, if not more than one, carbon-to-carbon double bond. And in fact, if there is only one, we talk about them as monounsaturated. Polyunsaturated, you've got more than one. And a lot of times you'll see those listed on food labels. Polyunsaturated is better than the monounsaturated. And unsaturated in general, health-wise, is better than your saturated fats. So what ends up happening here are things like your plant and fish oils where you get this kink. And so they're oils, they're liquids at room temperature, which also is what sort of helps them to be a little bit more heart healthy for people. Alright, those saturated fats can also, because they're solid at room temp, kind of get stuck, get cloggy, get sticky in your arteries in your body, and that's what leads to some of that heart disease, where these guys being oils are going to move through those arteries and veins much more smoothly. Phospholipids. So still have that glycerol. Alright, and if I take a look here, it is. Now they've only got 2 fatty acid chains, and that third carbon is now actually attached to a phosphate group. So these are, we talked about them when we were talking about water. These make up your cell membrane, that bilayer, right? So you've got these phosphate groups, these heads that like the water that's outside and like the water that's inside your cells. And then you get that double layer because those fatty acid tails can then get protected on the inside of that double layer and not have to interact with the water because they are, by definition, hydrophobic. Last type of lipid that I didn't mention to you earlier are these things called steroids. And the reason I didn't bring them up before this is because they don't have a glycerol fatty acid structure at all. In fact, they are carbon skeletons that have these 4 fused rings, and the ring structure in terms of how many double bonds go on the inside can be a little different. The groups that stick off can also be a little different. Why do they get thrown in here with lipids? Well, they're biologically necessary, so we had to put them in a category somewhere, but they are also hydrophobic, so they don't fit that sort of structure of anything else, and so they end up in the lipid category. Why are they important? Well, let's talk cholesterol as a simple example. So cholesterol is a steroid. Most people hear it in terms of you've got too much cholesterol in your diet, right? And so it ends up causing, once again, heart disease. But there is a difference between good cholesterol and bad cholesterol. You do need cholesterol in your body. In fact, it's a major component of your cell membrane, just like those fatty acid tails and phospholipids that we see are. But cholesterol in your cell membrane actually helps regulate the temperature. So if you're getting kind of warm, it's going to keep your cell membrane structure intact without letting it kind of ooze all over the place. Flip side, if it gets too cold, it keeps it fluid enough, at least within reason, to still let things pass in and out. And it's also used in your body as a precursor to create other biologically important steroids, a lot of which are things like testosterone and estrogen, which are your sex hormones. So sort of the building block that we then build off of to make these guys, which is also necessary for your body to function on a regular basis.